Hello everyone, welcome to today's Crown Bio Tech Talks. I would like to take this opportunity to present our SynAI platforms. The SynAI platform is a in silico compound synergistic effect prediction solution which belongs to the Crown Bio Crown Sync drug combination analysis service. Today's section is divided into 4 parts which answer the following questions. Why in silico synergism prediction is interested in the first place? What our SynAI platform can do for our customer. We will go further down into the technical details on how SynAI is built and how well the SynAI actually performs. In the following sections, we will provide the answers to all those questions. We will starting our first questions. Why in silico synergism prediction is interested in pharmaceutical researchers in the first place? Synergism effect or combination effect is a well-known phenomenon which the administration of multiple drugs together can actually create a greater treatment effect compared to a single drug treatment. In clinical practice, often multiple standard of care packages are combined to increasing the treatment potency. A synergism effect may reduce the drug resistancies or the toxicities during the courses by simply reducing the dose required for each individual drugs. Therefore, to understand the potential of synergism is an important step in the early stages of drug development. The assessment of the synergism is often accomplished using ex vivo's in vitro screening, which can quickly become expensive to perform with a large amount of candidate. At these steps, SynAI provides a pre-selection solution which can reduce the amount of potential candidates for the later screening process. Thus, to further reduce the screening cost, meanwhile, the screening procedure will also become more efficient with merely a smaller set of candidates. Now, let's first look into the web interface of SynAI. The web interfaces of SynAI is designed to be self-explained and intuitive. Actually, it's very straightforward and allows me to walk you through the platforms. On the left-hand side, we have a very neat input area where the cell lines and the drugs can be selected, or of course, customized drugs can be inputted using the smart sequences. And more than one prediction can be performed simultaneously. The user only needs to add multiple entrances for the designated combination. Even with the same combination of the drugs, you may choose 10, 20 different cell lines for the predictions. And on the right-hand side, we can see the output of the predictions where the name of the cell lines, the name of the drugs, and the blood scores from the predictions will be elaborated. And the table can be downloaded, of course, in a standard CSV format. But compared to existing platforms, the SynAIs not only focus on the accuracy, but more focus on, as an industrial solution, more focus on the reproducibilities and the repeatabilities of the results. And from the highlighted result, we can immediately see that the orders of the compound is reversed. And in SynAI, the AI course is trained to force the scenario where the input orders of the compound will not affect the outcomes of the blast. So for the same combination of the drugs, it doesn't matter if it's compound 1 or compound 2 or compound 2 versus compound 1. The blood scores that from the prediction will stay the same. And the second point is that we focus on the prediction, or we try to increase the sensitivities of predictions to the highly potent combinations. And in contrary, we are trying to reduce the sensitivity to try to create a false positive from a marginal cases. This is important in the candidate selection because obviously we can just say every compound is sensitive. But then what's the point of having the entire collection pass on along for the next step screening if there is no real selection process has been performed? Therefore, the ideas of SynAI again is to emphasize the fact that the platforms can reduce the candidates for the later in vitro ex vivo screening process. If we look further down into the input areas, if you look into the later websites, there are actually 4 modes provided by the SynAI platforms. The default mode is known as the drug versus drugs mode, where the user can simply select existing cell lines and the compounds from a drop-down menu. This is not quite useful if you do have a customized research question, but more like for benchmark studies and for evaluations of the platform itself. The more interesting versions of the modes is the drug versus SMILE sequences in the SynAI platforms. In this particular mode, the user can actually choose the different cell lines and give the customized SMILE sequences as inputs, and then select any existing compound which has been tested or known to our database. This is particularly useful if the customer is providing a small molecule engineered compound which want to test or evaluate the potential synergism effects for the compound crossing a standard of care packages in several well-known cell lines. Now, of course, the most flexibility is provided by the third mode, where both of the inputs is actually replaced with a SMILE sequence inputs. The user can simply just provide the SMILE sequences as much as they like and separate it using a new line and choose the cell line, and then the prediction will be performed on the designated cell line crossing all the user input SMILE sequences. This is what we call the free mode, where the user can just simply try whatever they want with their compound candidates. To further simplify the process, one of the customers actually recommend that maybe we should have a fourth mode where you just simply have one input area for the SMILE sequences, where the sequences are separated using new line. So the user can simply just dump in the entire collections of the SMILE sequences and choose the cell line for the prediction test. And then the tester will simply just perform a crossover comparison for all the SMILE sequences that has been provided in the area. Although so far we don't quite understand why this is needed, but as the user is actually very interested in this particular mode, so we actually have this one as a fourth option, as a simplifications of the third mode, which is the SMILEs versus SMILEs models. And we believe that in most of the cases, the third mode, the SMILE versus SMILE mode, will be the most popular interfaces where the user will entering our systems. Now, of course, you're welcome to give a try on the SynAI web interfaces already from the website provided. But let's not be so hasty, and let's look inside the SynAI and try to see what makes it ticking. The typical type of biological research questions such as synergism prediction can be easily converted into machine learning questions, normally known as the regression modeling. In simple terms, we have drugs information, we have synergism information under designated cell line. We want to build a computer programs which is capable of predicting the synergism using just the cell line information and the compound information. So how do we starting with it? Of course, with some existing viabilities and toxicity assay data that are drugs or compounds are being tested with different cell lines, and their synergisms has been measured and converted into blood scores. For the drug itself, we also need some representative information. In this case, we choose the most basic information, which is the SMILE sequences. By combining these two data sources, we convert first the SMILE sequences into molecular fingerprints, which is aggregated characteristics and abstract representatives of the molecular compound and its structures signatures. Here we choose the PopCam fingerprint and the Morgan 2D fingerprint. Later we will explain the motivations in the hyperparameter tuning studies. But for now, as you can see, that molecular fingerprint is a form of hot encoded systems which represent a different biochemistry phenomenon. We're not going to dig too deep down into this. But for examples, here we have maybe a one which suggests that this particular compound has more than 4 hydrogen bonds, etc., or or has a certain presence of a metal components, etc. And for the neural network training, the molecular fingerprint will be used as the input dimensions. Of course, with the input, we need a output dimension, which in this case is the blood scores. So the trainings of the neural network is essentially try to numerically build up a correlations between the inputted molecular fingerprints and the blood scores. So somewhere in between, there is a lot of neurons which has tons of parameters, which mathematically convert the input into the output. And during or through these procedures, we'll be able to use only the SMILE sequences, convert it into molecular fingerprints, combine the features, and then finally predict the synergism scores of the combinations of the compound under certain cell lines. In our cases, here we see a typical examples of such combinations of the features. If we further look down into the data sets that used for the AI core trainings, there are basically 3 data sources that has been employed. 2 of them are public and well-known data set which has been repetitively mentioned in existing literatures. Of course, one of the private data sources are coming from CronBio's HuBase. The NCI dataset is a very well-known Synergism dataset where it consists of 60 NCI cancer cell lines, which has been tested crossing 100 FDA-approved small molecule treatment solutions. As a result, each of the cell lines will have roughly 5,000 to 8,000 pairs of compound readouts with the blood scores and the compound. The second data set is the drug combo DBs, which is a newly generated data sources. It does itself generate some new toxicities or viabilities assay data, but the majority is a combinations of all existing data sources. So it's more like integrated solutions crossing multiple data sources from all kinds of publishers. So underneath, we can see examples of such data, how such a data looks like in the actual training process. So we have the drugs names, we have the SMILE sequences, we look up from the PopCams. That is done, of course, automatically. And also the cell lines, which is usually coming from the original data sources, and the blood scores that has been measured using either viabilities or toxicities assays. The trainings of the neural networks of SynAI is actually straightforward. Here we starting with a n-fold cross-validation strategies for the buildings of the neural network itself. First of all, the data set is divided into a working set and a testing set, where the testing set, as you can see from the diagram, is being left out of the entire training process and used as an independent testing set for the final performance of the networks. The working set is divided into 10-fold, where during each of the iteration, 1 fold is actually left out as an internal evaluation set, where the remaining 9-fold of the data is used for the buildings of the neural networks. And the performance of the neural networks during each iteration will be evaluated using the so-called left out fold, which is sometimes referring as the validation sets. Now, the same procedures will be repeated 10 times, And during the repetition, the networks that produced the highest performance based on the validation set will be chosen as the final networks, which the final performance will be then evaluated using the best networks versus the testing sets. Now, of course, here comes the enforcings of the input order strategies. So instead of just using the original data entrances, which normally looks like compound 1 and compound 2, you have a black scores. Here we duplicated every single data entrances with a secondary clone entrances, but the molecular fingerprint is actually generated from a reverse orders of the compound. So if the original data input is compound 1 versus compound 2, the clone set is actually compound 2 versus compound 1, but both with a equal black scores. The problem is that with such a duplication, it actually increased the complexities of the training processes for the neural network, and it makes it even more difficult to build up the neural networks. From the initial trainings, we immediately noticed that the parameters or the complexities of the neural networks, or the choices of important features like loss functions or optimizer, can greatly impact the output of the prediction accuracies. From these slides, you can see the actual iterations of epoch during the training, where different loss functions and different optimizer has been selected, and a different neural network architecture has been used. And in these cases, we do want to have a high accuracy for the prediction, but we also trying to avoiding the introductions of overtrainings. Therefore, we want to select these parameters, or these hyperparameters, in fact, in a more objective fashion. This is where the hyperparameter tunings actually comes in. The hyperparameter tuning studies is the procedure to select the most suitable hyperparameters for the training process of the neural networks. We're not talking about the parameters inside the neural networks, like what is the parameter for each of the neuron, but rather we are actually looking into the hyperparameters. For examples, in this particular cases, we're talking about what is the learning rate we should be using, what is the maximum epoch. If you're familiar with the PyTorch, the PyTorch packages, you probably know the meanings of these parameters. But also, of course, what type of the network will suit the best. We have the more standard linear networks, but we also have the more complex, the convolution networks. We also have the recurrent network. So which one will work the best? Which one will actually produce the best results? And also even for the same type of the networks, we have to decide how many layers would you like to use. The number of the layer increase the complexities of the network, but also increase the chances that the network may produce a overtraining results. On the other hand, the trainings of the neural network is itself a optimization procedure. So you have a choice of, you need to make a choices on the loss function and optimizer that will be used for the training procedure itself. Now, on top of everything, we mentioned this earlier in the early slides, is that which molecular fingerprint actually will be the best for the prediction process, because it's more than just one choice. We already have like 3 different choices for most of the analysis. We have the PopCam, we have the Morgan, we have the Mac fingerprints. So which combination actually produced the best predictions? Because the more information, sometimes we know that the more information is actually equal to no information. So actually providing more information does not necessarily lead into better predictions. But these kind of testing is very, very laborious. So for the test that we run, actually it took almost 10 days with GPU supported cloud computations to actually finish just one tuning, just one hyperparameter tuning for one single cell models. So this is a huge amount of work, but we want to emphasize that because as an industrial solution, we want to see that our solution is stable, reproducible, the prediction result is reproducible and is repeatable. That's actually the most important goals here. Accuracy is one thing, of course, everybody wants to have high accuracy, but the stabilities of the platforms and the reproducibilities of the results is what we always looking after. Here is a coordinate plot chart, which is a popular visual representation of how a hyperparameter tuning result looks like. Each of the columns represents one choices of one hyperparameter value or choices here. So as you can see, some of them is discrete, some of them is continuous. And each of the line representing a combinations of choices. For example, if we follow one of the line, is starting with a mean square errors, which is a common choices for regression studies. And we have a ADAM optimizers and using 8000 as the maximum epoch for the training, and the learning rates around -4, etc., etc. So the result actually representing the combination of all the tests that has been run for a single cell lines. And we can see the training PCC and the individual independent testing PCC from the Pearson cross correlation from the regression results. And as we can see from this particular cases, the numbers of the hidden layers for the neural network actually matters. And when it comes to a simple networks, which only was one layer, the prediction output is always a little bit relatively lower. Where when the increasing of the complexity goes up, it actually increased the performance. So how well exactly is SynAI perform crossing the NCI datasets? Well, fortunately, we have some existing literatures using the same datasets for synergism predictions as well. And from the comparisons of the outputs, our SynAI predictions actually produce a relatively similar and slightly higher performance compared to existing literatures. The reason of not getting any higher, I mean significantly higher performance, is actually because of the NCI later reports the dataset has a internal consistency across consistencies lower than 0.71, which means that for the replicates of this entire experiment, the internal consistencies crossing the replicates from the same conditions and same cell lines are roughly around 0.7, which actually suggests that it is impossible to outperform such numbers with the regression prediction or with the predictions of the BLET scores. So we are actually happy with the performance because it just cannot get any higher. But on the other hand, the performance that we yield in our cases are much more stable. If you look into the charts on the back, which is the validation PCCs of our crossing the entire NCI 60 cell lines model. Some of the models we can even yield prediction accuracies around 0.9, I think, almost 0.85. While some other models which are producing relatively lower accuracies. Later, in the studies, further studies looking into the individual cell model, what we realize is actually because those particular cell models are in general just less potent to all treatment. So it's hard to say whether if they actually respond to any kind of treatment. So they're just not really sensitive to all the treatment that has been introduced, which are sometimes like triple positive breast cancers, or so those cancers which are difficult to suppress, are usually produce not very clear data anyways. So the actual outcomes of the experiment, I mean the lab experiment itself, is also quite inconclusive. So in that sense, we can say it is also a certain phenomenon which is interesting to actually follow up. I'm sure there has already been quite extensive amount of information which we need to digest it today. So at this point, I would like to conclude our today's talks with a rituals of the value adding of our SynAI platforms. First things first is the learnabilities of our platforms. The AI cores of the SynAI platform is retrainable, which means that any new cell model data or new compound data can be fed back into the platforms to allow the AI to learn new possibilities of predictions. Of course, the reproducibility of the platform is guaranteed and enforced by the training procedures, by the rules that we employed in the training procedures. Finally, we stick to pure SMILE sequences inputs to provide the maximum flexibilities for small molecular engineering, and especially there is no need of compound synthesization for the prediction procedures. It's just pure text-based informations. Of course, this is not yet the end of our development. Our latest progress including using graph-based networks and more cell model data to expanding the functionalities of our SynAI platforms. During the studies, we also realized that the applications of similar principles can be employed in yet another important studies in the pharmacokinetics, which is the toxicity studies, or the ADMENT predictions. Our latest development in the new systems, which names as the ToxAI, is a platform that allows the predictions of toxicities using the SMILE sequences. And I'm sure that in the later text talks, it will be also drawing your attentions. And follow the links. You can find the SynAI platforms and the further informations of the Kuan Yan services for the drug combination analysis. Please feel free to have a quick look into the links. With these information, I would like to thank you for your times to join our text talks today, and I would like to invite questions. Thank you very much.